This Week in Virology, the podcast about viruses, the kind that make you sick. From Microbe TV, this is TWiV, This Week in Virology, episode 620, recorded on May 27th, 2020. I'm Vincent Racaniello, and you're listening to the podcast all about viruses. Joining me today from Austin, Texas, Rich Condit.
Hi there, Vincent. How are you doing?
From meeting to meeting.
Yeah, it goes like that. So we got 85 degrees, partly sunny, partly cloudy, headed for near 90, slight chance of rain. It's a pretty typical, we're running up to summer Austin day. And
from Madison, New Jersey, Brian Barker.
Hey, it's great to be here. We have 79 degrees and partly cloudy here.
And returning today for another bout of immunology from NIAID of NIH, Jon Yewdell. Welcome back.
Hey, guys. Great to be back. What do we got? We got 78 degrees and cloudy.
And you're still home, right?
I am still home. I haven't set foot in the lab in probably 6 weeks or more.
Wow.
We're allowed to have one person per bay. So I've got 4 or 5 people who can work at any one time, and they're arranging their schedule. So they're obeying social distancing and they're wearing their face masks. And
you periodically talk to them, see what's going on?
Yeah, all the time. This is for you guys, for everybody, for all of us. I mean, the Zoom is a game changer in all the other ways we can talk to each other. So yeah, we're doing a journal club online. We just had ours today every week, and we have our lab meetings. And, I think after this is all said and done in a couple of years, there's probably going to be a lot less in-person conferences and meetings. And we'll have to reevaluate everything we do in science, particularly in terms of the travel.
Yeah, I think that's going to be the biggest change is business travel.
Yeah.
Interesting to see how what happens.
And just from the climate change perspective alone, is it really worth, most of us scientists, we do tens of thousands of miles, if not more, per year flying. And we are all professed to be green people, but there we go. I mean,
yeah.
It's a very selfish thing, really, and I'm to blame as much as anybody. And so I think, there will be positive elements that come out of this this global disaster.
Well, if that's true, and I think you might be right about the reduced travel, then a lot of businesses are going to cut back. Airlines, hotels, restaurants, right?
Yep.
The economy is going to take a long time to reach a new equilibrium.
Yeah.
But people have been agitating this for decades. Actually, is that all our measures of successful societies are based on this kind of GDP measure, which is probably not completely useful for humanity. Mm-hmm. And, we just really, particularly with global climate change, which in the end is a much, much bigger problem than COVID-19, this is the time to get serious about this now. I mean, this is a warning shot over the bow of humanity, right? This is what nature can do to you. And we're idiots as a species if we don't take this opportunity to rethink what we're doing and make changes in the positive area. And let's face it. I mean, as rough as this is on the wealthy countries in the world, it's going to be way worse on the poor countries of the world. And that's something else that needs addressing, not just the wealth disparity within our democratic societies, but the wealth disparity in the world, if we're going to live together.
All true. I agree with all that.
Well,
so not much to do about virology. So let's get to it.
All right. Thank you, Jon, for coming back. You had a lot of great reviews last time. People loved the explaining immunology. So I thought we'd have you back to talk about a couple of papers dealing with immune responses. And first one, let's do Tcells first.
Okay.
The paper is in a pre-proof to what journal is this? Cell. And it is called Targets of T-Cell Responses to SARS-CoV-2 Coronavirus in Humans with COVID-19 Disease and Unexposed Individuals. You must recognize some of these authors.
Yeah, no, we had a very close relationship with Alex Sette over the years and Bjorn Peters. And this is the foremost group in identifying and the mass scale of peptides, not just for viruses, but for other pathogens as well. And they also are the authors and the maintainers of a wonderful website called the Immune Epitope Database, which is a fantastic resource for the immunological community for discovering new peptides and the proteins you're working on, and also in cataloging everything that's been published and known about T-cell, particularly T-cell epitopes. The B-cell epitopes are much harder to catalog because they're three-dimensional in their nature. But the T-cell epitopes, of course, are perfect because they're basically just digital. So this is a premier group, probably the best group in the world in doing what this paper is about.
Just to fill out the authorship, the first author is Alpha Grafani. There are about 21 authors. And there's what I would say 2 senior authors at the end of the authorship that are asterisked as making equal contributions, and those are Shane Crotty and Alessandro Sette.
Yeah, Shane no longer is a rising star. He is a risen star in the viral immunology field, mostly B cells. But Shane knows a lot about a lot of things. He did his postdoc with Rafi Ahmed. And actually, Vincent, you probably knew him. He had this wonderful paper on that was Shane's PhD thesis, was about how ribavirin works with poliovirus.
Oh, yeah, sure.
With Raul Andino. It was a
good stuff.
Great paper. Yeah, we interviewed him for a postdoc, and he wisely chose Rafi Ahmed's lab, but he would have been great. He's a really good guy. Actually, as an undergraduate, he wrote a biography of David Baltimore.
That's right.
Yeah,
I have it right here.
He's a very talented guy. Yeah, Alex is also very interesting. You know, it's the great thing about science. You get to know people, and Alex he's a fencer, and he's been in a number of operas out on the West Coast as a stand-in. He's a very interesting guy.
Shane and Alice had a foray into poxvirus transcription at one point. And so Shane and I intersected over that. And by the way, this is coming from the Institute for Immunology in La Jolla.
So Shane actually sent me this manuscript a couple of weeks ago. And this is happening more and more. People are sending TWiV their papers, So I said, well, we'll get Jon to come on and explain it. So, Jon.
Well, this paper is fascinating, even if they hadn't sent it to us.
Yes, of course.
Yeah.
So, Jon, what is a T-cell epitope?
Yeah, so we have 2 flavors of T-cells. We have CD4 T-cell, the helper T-cells, and the CD8 T-cells, the killer T-cells. And the CD4 T-cells recognize a molecule known as an MHC class II molecule. This is a molecule that binds peptides of at least 10 or so residues. It doesn't care so much about the ends of the peptides. And so it basically, when something like a virus is internalized into an endosomal compartment, the endosome, as it progresses into the cell, becomes more acid. And enzymes get activated and proteins get activated that work to unfold the proteins that are coming in and to cut them into bits and pieces. And the MHC class II molecules start binding the unfolded polypeptide chains as they are unfolding. There's trimming that occurs within the groove. These molecules are then sent back out to the cell surface with a peptide, probably who knows what the real length is, but around 15 residues. And these peptides then can activate the CD4 T cells, which will play a critical role in the lymph node in the differentiation of the B cells. The B cells are the cells that are producing the antibodies. A B cell without a T cell can only make an IgM antibody response that is low affinity and also very transient. For a antibody response to become an IgG response, it has to have T cells. So this is important for a lot of what you guys have been talking about, because if you have a good IgG antibody response, it means you have a good helper T cell response. You cannot have an effective antibody response without a helper T cell response.
So importantly to me, I want to get this straight. The process you described says that these CD4 T cells are the peptides that they are presenting arise from proteins that were taken up by the cells, not necessarily proteins that were synthesized in the cells. Is that correct, or is that an overstatement?
Well, no, that is correct, but I didn't fill it in and you misstated, Rich. It's not the T cells that are doing it. The T cells are waiting for that to be done.
Okay.
T cells are then recognizing the APC, the antigen presenting cell that has done that. Now, in a naive individual, the antigen presenting cell is probably going to be a dendritic cell. This is this specialized blood cell discovered by Ralph Steinman that resulted in winning the Nobel Prize for. And these cells are really, really good at activating T cells. And so in the absence of a primed immune system, they will be doing the presenting. If the individual has been exposed to the antigen by vaccination or by infection, then the presentation actually, and this gets tricky, and so we can slow down here, it gets presented by a B cell that has the receptor for the antigen. Okay, so once we are infected with coronavirus or vaccinated, we will have B cells that recognize the surface proteins of the virion, particularly the spike protein, and this will give the B cell a huge advantage at antigen presentation, because no longer does the antigen presenting cell have to find a way to stick to the pathogen to present it. Now, because of this high affinity receptor, and then a super high avidity, because it's the whole surface of the B cell that can be mobilized to collect the antigen, the B cell will bring the antigen in and process it. And B cells are very, very efficient antigen presenting cells for CD4 T cells. And remember, the whole immune system is designed anatomically to present antigens. The lymph nodes are where the lymph comes to the lymph nodes. And so the B and the T cells are intimately exposed to each other. And so the initial priming will go by a dendritic cell, but then once the immune response gets going, you're going to get presentation in the lymph node, even in a previously naive individual, someone who's responding now, where the B cells are presenting the antigen to the T cells. And then within the lymph node, the B and the T cells are going to be setting up a competition between the B cells, so that the B cells that are most able to present antigen to the T cells will be the B cells that produce the most progenitors. So as we talked about the last time I was on, it's this incredibly cool microevolution that goes on within the lymph node. So Darwinian selection is occurring in real time, people, listeners, as you are infected with the virus. And the two kinds of Darwinian selection, of course, the viruses are always being selected for what replicates the best and the fastest, but the immune cells are also being selected for what's best. And so the B cells are competing with each other, and the competition for the B cells is basically who can get the most help and therefore beat the other B cells. And through that process, you get what's known as affinity maturation of the B cells, where the antibody sequence is actually changing all the time. And we'll talk about that in one of the papers that's coming up, so we can wait on that. But the T cells are what's basically driving that B cell evolution. So that's the helper peptides then are the peptides that the T helper cells are going to recognize.
So for me, oh, sorry, go ahead, Brianne.
Oh, I was just going to sort of say, Rich, usually when I think about antigen presentation on MHC class 2, at least sort of the classical way of thinking about it is exactly as you said, with a protein that has been taken in, internalized either by endocytosis or phagocytosis in the cell. And if you look at some of the sort of simplified versions of textbooks of this process, they talk about it as being the exogenous antigen presentation process, because it does involve these antigens that are being taken in by the antigen presenting cell.
Right. And for me, this is, I'm constantly tinkering with this because it has important implications for vaccination to me.
Absolutely.
Because some vaccines involve injecting the antigenic protein. So that would be an exogenous antigen, whereas others involve synthesizing the antigen in the cells on site. And my understanding, correct me if I'm wrong, is that the peptides from those two different delivery methods, if you like, of antigen, or they're going to be presented on different MHC molecules and shape the immune response differently. Is that correct?
Yes and no. Yeah. I mean, it's complicated because there's a lot of overlap. So the Venn diagrams overlap considerably. So let's say we have the Moderna mRNA vaccine. Okay, because there's been a lot of talk on the show, and that's probably the first one that's either going to work or not work that we're going to know about in this country. Okay, so the Moderna vaccine is this mRNA vaccine. So you guys have talked about that. This is the messenger RNA and encodes the S protein. That's what's in the vaccine. They inject this into the muscle, and in the muscle, probably what happens is even though the muscle cells can be transfected by this mRNA because of the vehicle it's in, probably the relevant cells for the immune system are still going to be the dendritic cells. That dendritic cells are in every tissue, and then once an inflammation gets going, they're attracted to tissues. And the dendritic cells are going to get transduced and start expressing the protein. And so in that case, these cells will have the opportunity to have both direct presentation, which means they've synthesized the protein, and cross presentation. Because what dendritic cells can also do is acquire antigens from other cells, Rich. So let's say we have a coronavirus infected cell or an mRNA transduced cell. It will make protein, right? So for that, it's endogenous. But if you're a professional antigen presenting cell like a macrophage or a dendritic cell, you can acquire the protein from that cell and then process it via the classical exogenous route.
Okay.
So for class 2, particularly, there are two routes that could happen, the endogenous and the exogenous. And then the endogenous route, then let's say the dendritic cell makes the protein. There's two ways that can work. The protein can find its way one way or another into an endolysosomal compartment and be processed in a classical class 2 way. And then there's a whole other pathway which has not received a lot of attention, but this is from the work of Lawrence Eisenlohr at the University of Pennsylvania. What Ike is his nickname, what Ike has shown quite clearly in the flu system in mice is that something akin to the way class 1 antigens actually works for a substantial number of the class 2 peptides as well. And that work, which is really spectacular work, it's never been extrapolated to other viruses. And what the contribution then is for other viruses, and particularly in the human situation, at this point has never been evaluated. But it's probably substantial.
Yeah, I think that Jon, you're much more of an expert on antigen presentation than I am, but I would say the one other piece to this, Rich, is that you might imagine that some of those transduced muscle cells might die. And those dead cells could be picked up by other cells by phagocytosis. Right. So we do see both CD4 and CD8 responses in nucleic acid vaccines, but you might imagine slightly different responses depending on the vaccine type.
Absolutely. And the contribution of the dying cells or even just the cells that are fragmenting versus the transduction of professional APCs from the tissues. In humans, it's not known because it's so hard to do the experiment. But even in mice, it's not easy to tease apart. And so the answers are really kind of conditional. We know these things can happen. And the extent to which they do happen is really not really known. And there's something else which is quite cool. I think I talked about it last time, which is called cross-dressing, which is when the professional APC actually picks up a preformed complex from a cell that's transduced or infected. Okay, so for class 2, that's not going to be relevant because cells do not express class 2 in a resting state like a muscle cell. But every cell will express class 2 when it's exposed to gamma interferon. And so as the inflammation is progressing and as you're getting NK cells involved in the innate response and CD4 and CD8 T cells are recruited to the infection site, all those cells, including the innate cells, will secrete gamma interferon. So class 2 will be expressed now in basically every cell that in the active infection site. So things are going to get really, really complicated.
Yeah, okay. I feel like I'm back at the lunchroom table in grad school having this argument with my colleagues.
Yeah, and it's just, we've published a lot of the papers on this about what could be working in mice, and basically everything you look for you can find. And so the relative contribution will vary between the different viruses, the different sites of infection, probably even the different proteins, and even at the level of peptides, there can be different responses. So to finish the question that Vincent addressed, though, what is the CD4 and CD8? So the other side of this are the CD8 T cells. These are looking for MHC class 1 molecules. Class 1 molecules are expressed by every nucleated cell and even by some non-nucleated ones like platelets, which actually have a lot of class 1 per unit volume. And what the class 1 molecules do is they find peptides between 8 and 11 residues in length and bring them out to the cell surface for immunosurveillance for the CD8 T cells, whose principal job is to kill infected target cells. So the original name for them was the cytotoxic T cells, but they also secrete cytokines like interferon and TNF that have other functions in the response.
So the peptides that are presented by class 1, what's their origin?
They're good question. So classically, they're endogenous proteins, and their origin in a virus-infected cell. And this has been, sort of the major one of the major focus areas in my lab for the last 30 years or more. Actually, I started working on this in the 70s, believe it or not. What happens with viral infections is as the viral proteins are translated by the ribosome, some fraction of the translation products are diverted to the class 1 system so that they are very, very efficiently presented to the for immunosurveillance. And, we've shown that you start to make peptides exactly at the time you start to make proteins, regardless of what the fate of the protein is. So for the class 1 system, you can have a peptide in a protein that's going to be secreted from the cell. So the cell will have no chance, in theory, of presenting the peptide. But in fact, secreted proteins are very good sources of class 1 peptides. And there was a theory we proposed in 1996 called the DRIP theory for defective ribosomal products. And the idea was that nothing is perfect, including translation, and that mistakes would be made, and that from those mistakes, you would be able to make antigenic peptides. And the exact details of the theory were not completely correct, but the idea was really correct. And that is that as you make proteins, you make other forms of those proteins that are then degraded principally by the proteasome, which is the major protease in the cell responsible for protein degradation. These peptides are then transported into the endoplasmic reticulum, where through a highly choreographed series of events, they are loaded onto MHC class 1 molecules for expression on the cell surface. So they move through the Golgi complex to the cell surface, and there the CD8 T cells can recognize them and kill the cell, very importantly, quickly before the cell can release progeny virions. So that's the whole point of the class 1 system. Viruses, as you guys know, are very, very fast and can go through an entire infectious cycle in 4 hours at the most extreme, and certainly within 12 hours. And so if a T cell is going to have a function at preventing virus release, it has to be a system where you could recognize viral protein expression as quickly as possible. And that's what nature has done. So the class 1 system is geared to this rapid peptide presentation.
So Okay, we have CD8 and CD4 epitopes, and they're different, right?
They can overlap, but they tend to be different,
Okay.
yes.
So in this paper, they identify them in convalescent patients. So how do you go about identifying these epitopes?
Yeah, so you get their blood, and you get the peripheral blood monocytes, and then you expose them to antigen presenting cells, which it gets tricky now. So this is really good. So why is it tricky? Why it's tricky is why Brianne just can't get Rich's liver and accept it, right? So the MHC system.
I'm not offering anyway.
Okay, how about a kidney, Rich? You got two.
Okay.
Kidney, maybe, yeah.
Okay, thanks.
Yeah. Okay, so the whole system was discovered basically because of this transplantation issue. So the MHC, what does the H stands for? It stands for histocompatibility, which is just a fancy word for tissue compatibility. And what that means is that we have these class 1 and class 2 genes, and they present all these different peptides to the immune system. And this system is not just turned on for viruses. It is constitutive, i.e. every cell in your body has class 1, and all of your professional antigen-presenting cells have class 2. So if I take Rich's liver and try to put it in Brianne, there's all these new peptides and class 1 molecules. And a different class 1 molecule looks like the best viral peptide that the system has ever seen. So what's known as an allomorphic difference between a class 1 molecule is recognized by 5 or 10% of the T cells. Okay, so they go absolutely crazy. And that is why tissue rejection is so potent, because you get this huge CD8 and also a CD4 T cell response against these MHC proteins. So what that means is that every person basically has their own custom immune system with a set of 3 different class 1 alleles, in other words, 6 potential molecules, because you have 2 chromosomes, 1 from each parent. So you have 1 MHC, like HLA-A, you have 1 from your mother, 1 from your father. Sometimes they're the same, many times they're not. So you can have 6 different class 1 molecules. You can have 6 different class 2 molecules. Class 2 gets even more complicated because each of them is 2 chains, and they can make hybrid molecules. So there's all sorts of permutations there. So basically every person has their own selective immune system at the level of peptide presentation, which means it's not just like an antibody where you can do one test that works for everybody. For the T cells, you have to have cells to stimulate the T cells that match at the HLA alleles. And that means practically that you basically have to use autologous, in other words, antigen-presenting cells from the same person. So fortunately, blood has not only T cells, but it also has monocytes in it and some dendritic cells. And so what you do then is, without really knowing what you're doing, is you take the blood, you expose them to either a virus, which they didn't do in this paper for good reason, or peptides. And then you expose the blood cells to the peptides. You incubate the T cells with the antigen presenting cells for a certain amount of time, and then you measure either the activation of the cells by cell surface markers, by flow cytometry, or you measure the cytokines that they secrete when they're activated. And you can do either way. So that's in this paper, I think they used the cytokine. I think they also used activation. If you know what the peptides are, you can do something much more specific, which is to make an MHC molecule that's already bound to the peptide, so you can quantitate the T cells and do it very, very carefully.
So when you're setting this up, do you actually separate out T cells from antigen presenting cells and then wind up mixing them back together, or is this all done on the mix to start with?
I have never done it that way.
So when
Brianne, you probably have.
When it was something that we did in graduate school, we would do it on the mix of cells. But I don't know if that's what they did here.
Okay.
We always did it with the mixture.
Okay.
I'm going to look it up while you guys talk.
Yeah, I'm looking too. I doubt they separated them. But
yeah, the separation process gives you a whole bunch of other potential issues, and then you have to think about.
That would mean that you've got blood, and that comes with a mixture of T cells and antigen presenting cells. And if you throw in peptide, that'll wind up being presented by the antigen presenting cells to the T cells.
Exactly.
So they say they just get PBMCs, which would have everything.
Yeah, I think that's what they did. And then a crucial part of this study, particularly, is that you can also freeze the PBMCs and then use them many, many years later.
I was wondering about
which is what they did. So that's really cool as well. So then you can go back into your bank and, particularly it's really nice. You can unambiguously know that people have not been exposed in any possible way to COVID-2 because you can get cells from before COVID-2 existed in the human population.
PBMCs for the uninitiated are peripheral blood mononuclear cells. And that's going to include all of the types that we've talked about. Is that correct?
Not so many dendritic cells, but it'll have lots of monocytes. Yeah,
lots of monocytes, lots of T cells, lots of B cells,
and then lots of granulocytes as well, which could probably present antigen.
So all of the experiments we're talking about can be done on samples that have been frozen away for a while.
Yep.
Okay.
Yeah, they can
many
be.
Times.
I'm sitting here thinking, I served 12 years in a biochemistry department and 25 years in a microbiology department, and I never took biochemistry. I never took microbiology. And obviously, I never took immunology. I've had to learn all this on the fly, and it's still going on. And immunology is particularly tough because it's really a moving target.
Yes, it is.
And you know what, Rich? As Jon said, it's complicated. That's what they always say. It's complicated.
Yeah. It is. It's really complicated, and there nothing simple, and it's not because we're stupid, although that's part of it. It's because everything looks complicated when you don't know anything. It really is complicated, and there are all sorts of processes that can occur in parallel. And again, it's one thing. Even in a mouse, it's hard to know what's going on. In a human, it's almost impossible to really know.
And nothing. It says I get the impression that nothing is really black and white.
It's not.
Right?
No, and nothing's very intuitive either.
Right.
Unless you've been doing this for quite a while.
So the question is, who would want to be an immunologist?
Me.
Yeah, actually, if I had to do it over again, this is something I might consider. This is really quite interesting.
Well, I think I think we're all immunologists now. Every virologist, because if nothing else, you're working with how the virus interacts with the immune system, and there's no way around that. So
what they actually did in this paper, they made peptides and put them in the cells that we're talking about, but they predicted what peptides to use, right?
Not really. I mean, they made pools of peptides. I mean, it depends. Some of them they predicted, and some of them they just made like all the overlap. I think for S, they just made this entire protein where they made this overlapping pools of 15.
Okay.
Mm-hmm.
Right. And so they did both. They always use pools.
As they say, they use this epitope database to identify peptides with increased probability of being T cell targets. But you're right. You could just make the whole, if you do overlapping 15 peptides, would you basically get every epitope that way?
Yes and no. I mean, so for the class 2, you would. For the class 1, probably not, because the class 1 peptides will not, they make 15 mers. 15 mers will not bind to class 1 at physiologic affinity. So for the peptides to bind to class 1, they have to be processed. And the way they did this, there's serum in all the incubations, and serum has proteases. Ironically, one of the major ones is ACE1, not ACE2, but ACE1, which is a very potent carboxypeptidase. And then probably ACE2 is as well. We never studied it. We are now. So we were the first lab to show that ACE could actually work on antigen processing inside the cell. Linda Sherman, an immunologist from Scripps, showed many, many years ago in the early 90s that ACE could exogenously trim peptides. And it turns out for class 1 peptides, that's actually probably critical to their activity in the way that this group has done this assay, where they take these peptides that are much longer than they should be and just throw them in on cells. And so when you do it this way, you are depending on exogenous proteases to create the peptides that are going to be recognized. And that is not a strength of this study. is the use of these very large larger peptides. But this, and the authors are quite honest about it. This is just the opening salvo. Mm-hmm. And under normal circumstances, this would have been the first 3 months of what would have been several years of work. Mm. Right. But, just you guys know how it is. I mean, the papers that are getting published now, even from very good groups, they're what used to be maybe 10% or less of a publishing unit.
For sure.
Right. And I'm not criticizing them. I think it's really important that this information get out there as quickly as possible. But the standards are not the same.
You're right.
And the level of certainty is not the same. And I think what they found is solid, but certainly it's going to be expanded on by this group and other groups in the future.
Yeah, they do a really nice job in the discussion of talking about some of the caveats to their work. A few of the big things I was thinking about when I was reading the paper are all discussed and mentioned there. So that was really nice. One thing that might be important to say here has to do with the sort of structure of how class 2 binds to its epitopes versus class 1. So class 2 is a little bit less picky in terms of its peptides. It can be a little bit more promiscuous with its binding, and the ends of the class 2 molecule are open, so the peptide can sort of hang out the ends. That's why the peptide length doesn't matter so much. Strangely enough, in every undergraduate immunology textbook that I work with my students, there is at this point in the textbook a picture of a hot dog in a hot dog bun. Where you see the ends hanging out of the MHC class 2. MHC class 1 has closed ends, and it's much tighter in terms of which peptides can fit in. And it also matters a lot more in terms of which specific amino acid residues are there. And so they talk about some differences in their class 1 and class 2 data in this paper. And I think that that's one other place where the difference is important.
So let me ask you this. They find epitopes or cells that respond to peptides for CD4, 70% of patients, CD8, 100%, convalescent. Is it surprising that it's not 100% for both?
Well, I'm sure it is. They just missed
Okay.
them. I mean, you can't make a IgG response without a CD4. And you always miss peptides. I mean, even in the mouse systems, we're still, even with flu, we thought 10 was going to be most of them, and it turns out it's probably closer to 50 or more, even in mice where there's much less allele. So this is just a start, and there's going to be 100% responses to CD4 and CD8. And if you restricted mice to PBMCs as opposed to being able to get spleen and lymph nodes, you would miss a lot of peptides. And it's not like these guys are unaware of that. I mean, so the peripheral blood, the humans are big, so you get a lot of cells, but you still don't get all the specificities probably. And particularly the way they've screened, they're going to miss them. So that's not, it's surprising that it's not, it's a little bit surprising that it's not 100, but I'm certain it's going to be 100%.
Yeah, Vincent, wasn't it the other way around? It's 100% of CD4s and 70% of CD8s.
Yeah, that's correct, okay, according to their graphical abstract here.
Yeah.
And that's that was where I was thinking about the fact that CD8s and MHC class 1 are a little bit pickier, so they might have missed some easy.
They're not a little bit pickier; they're way pickier.
Yeah.
You're right.
No, that was an underestimate.
Yeah, they're way pickier. Yeah.
So they found epitopes in most of or many of the viral proteins, not just spike, right?
No, epitopes are everywhere, basically. And they're randomly distributed throughout the proteome, basically, and of everything. And, for class 2, here it really would depend on what the relevant form of the antigen is. Because for class 2, if it's mostly the particles that are being presented, then it's going to be heavily biased to the structural proteins, i.e. the proteins in the virion. Right. And so that's from the reactivity, actually, one could predict what most of the presentation is coming from, whether they're focused on the structural proteins of the virion or whether it's just everything. And then if it's everything, then, it's both the virions and then also what's in the infected cells and that are being presented, as Brianne was saying, from when the cells are dead and dying. And all those will absolutely contribute. So I guess where the money figure then is figure 6, right? Because that's where they have all the individual patients with each of the known open reading frames. And something else I'll mention, although it's not completely relevant or terribly relevant here, but the first protein where they've done ribosome profiling on coronavirus 2 infected cells is out. And as one might predict, there are all sorts of unanticipated open reading frames. And these we know are very good sources of peptides as well. So in addition to what's the defined annotated open reading frames, sort of standard ORFs, as we would call them, there are going to be all sorts of non-traditional ORFs that are sources of particularly class 1 peptides. These non-standard ORFs, the proteins tend to be very short-lived, and that's a rule that is pretty consistent, is that the more stable proteins will be better class 2 antigens. The less stable proteins will be better class 1 antigens. So that kind of also holds. But no, there's nothing really surprising here, I guess. Nothing really surprising, Vincent, in terms of that. Maybe the absence of responses in some, but again, it's a question, I think, of how carefully it's been done.
I mean, they do find some epitopes in unexposed individuals, right? 40 to
now that's more interesting.
40 to 60%.
Yeah. So we've looked in my lab. We haven't published anything, but we've looked at the sequence conservation between the seasonal coronaviruses and particularly the beta coronaviruses and SARS-CoV-2, and there are some highly conserved regions. I don't think there's a single identical match in sequence. Now, T cells do not require a perfect match. There can be a lot of plasticity at particularly the residues, particularly the residues that are pointing down, right? So, as long as the peptide binds to the groove, what the T cell is going to see then is mostly the residues that are pointing up towards the T cell receptor. So if you have conserved substitution, so the peptide still binds to class 1 because you substitute, say, one hydrophobic residue for another one, and the residues pointing up are the same or similar, the T cells can certainly cross-react. So whether these are true cross-reactions or not, I'm not convinced. Okay. Because what you would really have to do is show within the pools what the peptides are and show at least that the peptides are conserved. If not, the proteins actually cross-react. So it gets hard because this is a BSL-3 agent. But, something they didn't do, which you would do right away if it was something easier to work with, for, say, you're looking for cross-reactivity, well, you would take SARS-CoV-2 infected cells and see if there were T cells from uninfected individuals to see whether they were turned on by those cells, right? Because that would really show you that the peptide was actually expressed and was relevant. Yeah. And there are all sorts of artifacts that can come from using peptides. There can be peptides that, when you use at high concentrations, cross-react, that would never cross-react when they're at lower concentrations. There are peptides that you can synthesize, but that the cell doesn't actually make, right? So what the algorithms are really good at predicting is what will bind to class 1 molecules in particular. And there you can predict, say, 80, 90% of the class 1 molecules that will bind. But the algorithms are not that good at predicting what a T cell can actually see as a foreign peptide. It's probably 50% of the peptides are invisible to the immune system because of tolerance. And then it also isn't very good at predicting what is going to be naturally processed and presented, because the processing machinery is not 100%. It will maybe a quarter of the peptides that could be presented are presented because the proteasome won't chop them, or other things happen. Other proteases destroy them, and so there's all sorts of mistakes you can make if you just assume that every peptide is going to be made. And these cross reactions, they look like something. I agree. But to really know that they were true, you would have to identify the peptides as a start. And then it would be really nice to show that at the level of natural processing and presentation, that there was cross-reactivity. And this is a really, really important point. So I'm glad you're honing in on this, because this could be one of the explanations for why you have different clinical outcomes in individuals. And it could be those that have pre-existing T-cell immunity do better, or ironically could do worse, because we don't really know what is responsible for that second phase of the infection, where you're really, which is the phase that puts people in the hospital and kills them, where you have this clearly overenthusiastic immune response. And there's nothing necessarily to suggest that it's T-cell driven, but we don't know that it isn't.
Mm-hmm.
So it could be, ironically, that if you have pre-existing T-cell immunity, you do worse. I doubt it. You're probably going to do better, but we don't know that. And, something you could look at then in the patients who you think you have, so you certainly could look at clinical outcomes, but you could also look at the vigor of the antibody response. And so, for example, if you had a pre-existing T helper response, particularly against the S protein itself, you might predict you would get a more rapid and more robust antibody response to S.
Yeah, that was actually kind of my biggest question with this paper, is that they used patients who were non-hospitalized. And I wondered what types of things they would see in the immune responses of more severe patients.
Yeah, that would be really interesting.
Yeah, it would.
So, John, these cross-reactive cells are CD4 positive T cells, they say. And not, so I assume that.
Well, I think they have both, don't they?
I know.
I'm looking at the graph. They seem to have.
They were both.
Yeah, I think they think they have both, but yeah, and again, I think it's really promising, and then you have to look into it and really define it. And we had one of the few papers on this, that a truly random cross reaction between peptides that are not related in sequence is really, really rare. And we actually did this with Alex 10 years ago, Alex's lab, where we looked at 7,000 different permutations of human presented peptides and different class 1 restricted T cells. We did this in collaboration with Andrew McMichael, who had a lot of T cell clones against HIV. And we showed we had one peptide out of all these 7,000, two peptide pairs that were a bona fide cross reaction of random peptides. So if you get a cross reaction, it's almost certainly based on the homology of the peptides. And again, it doesn't have to be a perfect match. It has to be a kind of if it's 8 or if it's 9, you probably have to have at least 7 that are identical. And then it can't be the sequence changes have to be conservative, but sequence changes tend to be conservative anyway because the protein has to function. So these could be real, but they would have to devolve the peptide mixtures and then go for peptide to peptide. And what they say, which I'm sure is true, is they just don't have sufficient sample size to do that at this point.
But we do know, my understanding from Stan Perlman is that in fact, previous infection with a common cold corona does not protect you from infection with SARS-CoV-2, correct?
It doesn't appear to. Yeah, but, again, we have all these different clinical outcomes.
Yeah.
And, and so what is that? And okay, everything's going to contribute. All right. There's probably genetic differences in people,
of course.
What the dose is, how exactly they got it. And just to reemphasize your point, there is no evidence that there are biologically significant strain differences yet in coronaviruses. It doesn't appear to be that. But the microbiome can influence, if we talked about last time, the microbiome can influence the outcome. But one of the things that really could influence the outcome is the memory response to the coronas. And, you do have immunological experience with a related series of viruses. So, it's probably not going to be a huge factor. And if it was a huge factor, everyone wouldn't be getting infected, right? So it really doesn't look like there are resistant people. But, again, it seems like we've been with this forever, right? But we haven't. I mean, we just have January to June is 6 months of experience with this virus.
Of course.
And, we know so little at this point. And so we really just have to have our eyes open and be looking for things, but just, be looking for the unexpected as well.
Okay.
So, John, one of the things, one of the reactions to this, that if I hear you correctly, is that the results in this are not too surprising. And so, and to me, one of the take-home messages is that you're getting, in a natural infection, a full spectrum immune response involving both CD4 and CD8
absolutely,
cells, et cetera.
Just what
is there, but and that's not too surprising. But this contrasts with the focus that you see in the literature and in the media on the antibody response, which we're going to talk about. Probably, it seems to me, because that's the easy thing to measure. Is there such a thing as an a natural immune response to an infection of some pathogen that is really heavily or completely skewed towards an antibody response? Or do you expect...
Again, you can't have an antibody response without a helper response. So are there viruses where you don't get a CD8 response? Interesting.
Yeah,
no, there's a really good guy named Lindsay Witten, who's at Scripps most of his career. And Lindsay says with, what virus is Coxsackie virus? So Vincent, like the enterovirus, right?
Yeah.
He says you get a really crappy CD8 response to Coxsackie viruses. And clearly you do in mice. And I don't know that that's been explained. And I'm not sure how well that's been looked at in humans. So it's possible there are some viruses there, Rich, that you don't get a great CD8 response to. And then, there's some ironic viruses like all the herpes viruses where you get this, like CMV, you get this amazing CD8 response, but it can't do anything because the virus has 10 genes devoted to hiding from the CD8 T cells.
Right. Okay.
Yeah. So speaking of which, it's always interested me that the little viruses, and this, even though this is a big RNA virus, it's still much smaller than the DNA viruses. They don't really, none of them yet. I don't think I'm missing any of them. They don't have any proteins that appear to have evolved to avoid CD8 or CD4 recognition, right? So I think, the strategy of these guys, HIV would be an exception. So that is an exception. But the strategy of these guys, I think, is basically, it's hit and run.
Yeah.
You get in, you get out.
Yeah.
Right. And so it's just like, okay, you have CD8 T cells, but that's not going to be a problem for me.
Right. We're going to outrun you.
I'm going to outrun you. Exactly right.
Or you take
and that's why, I don't think it's just that antibodies easier to measure, Rich. I mean, I think in terms of protecting you against infection, antibodies are clearly better at that.
Okay.
Right. And they're because CD8 T cells, even now that we know about resident memory T cells, CD8 and CD4s that are living in the mucosa, even those cells cannot prevent an infection by themselves, right? Because they can only respond once a cell is infected. In theory, the antibodies can do that. In practice, as we talked about last time, they don't actually do that. I mean, even if you have sterilizing immunity, you probably still have some cells that get infected. But I don't think it's really a bias that the antibodies are better at protecting you. I think they really are better at protecting you. And in most mouse virus systems that people have studied, they're better. But in many of those systems, neither of them alone is enough. That you need both an antibody response, which again always means a CD4 response. So they are always there. But you also need the CD8s in many of the mouse models to get complete clearance. Okay, so probably that's what's going on in humans. And, the immune system has gone to great evolutionary trouble to make this system. So probably everything's important. And nothing is really completely redundant here either when it comes to the adaptive immune system. They're probably working in concert with each other. Actually.
I would add hepatitis C virus, which changes the CD8 epitopes too. Right.
Oh yeah. So once you have a chronic virus, then you get all sorts of immune escape. But also, there's so good point, Vincent. Even with a virus that can change very readily, there isn't very strong evidence that immune escape is important to escaping the T cell response, and it's very hard to escape a T cell response. And one of the arguments for the whole evolution of the MHC polymorphism is that this was driven by viruses. In other words, so a virus can't infect you and escape and then do the same trick to me as I've got these different class 1 molecules than you do. Okay. So that was pretty clearly true until these tumors, these transmissible tumors got discovered, like are killing the Tasmanian devils.
Right.
And then you had to rethink, because everyone thought it was a joke that, transplant was going to kill you. It's no joke. I mean, there are actually, a number of organisms now where there are very lethal tumors, at least one mammal where there's a lethal tumor, which are the devils, where you could argue that was what was driving the diversity of the class 1 and the class 2 molecules. So, before that, we were pretty sure it was the viruses, but just goes to show you, what do you know? And it could still be the viruses.
Okay. Let's move on to antibodies.
Antibodies.
So we have 2 papers, and first one is from a group all over the New York City and Australia. Geez, so many authors. First author, Fatima Amanat, and the last author, Florian Kramer from Mount Sinai, and a bunch of people in between, and a lot of names I recognize. They made a serological assay to look for antibodies. And I don't really want to talk about the assay so much, but the results. But this is an ELISA-based assay where I think they make the viral spike protein, they stick it on plastic, and they ask if you put sera on top of it, do you get binding? And so they didn't have a lot of patients. They had, I think, 3 from New York and 12 from somewhere else. And these are all convalescent sera, and they have antibodies that react with the S in this ELISA format. And what I really am interested in, 2 things. First of all, they did some neutralization tests using pseudotyped VSV with the spike of SARS-CoV-2. And they say ELISA and neutralizing activity correlated, but I can't get a sense for exactly what the levels of neutralizing antibodies were. In the next paper, we'll get a better sense of that. But they tested 21 batches of pools of human immune globulin, which are usually given to people, from 1,000 donors each. None of them had antibodies to the spike. And I was wondering what you thought about that.
Well, yeah. So we talked about that last time. I mean, they all had antibodies to the spike. They just didn't bind very well. So, it's not all or none with antibodies, right? And so you can look at the data in the paper as well. I mean, you do an ELISA curve, there's always some binding if you start with a high enough, low enough dilution of sera. So, yeah, I mean, the assay looks good. I mean, and this is an assay, not to take anything away from these guys. It's a really good group. It's an assay that, a 10th grader could do, right? It's a very simple assay. The hard part of the assay is to have the antigen, right? Is to have the antigen in a form that's really native. And that's, data look good. It's really hard to judge, assays like this unless you have a panel of defined monoclonal antibodies. Because without that, how do you know exactly what you're measuring, right? And so even though you have something that it's receptor binding domain, great, that's good. But in an assay like this, you don't really know what fraction is native and what fraction isn't native. And I've done a lot of assays like this, and with a really well-defined panel of monoclonal antibodies. And basically, whenever you bind a protein, even if it was like perfectly native to start with, to plastic, some of it semi-denatures, which is not the worst thing, actually, because then you're exposing other epitopes that may actually be relevant to how antibodies work in vivo. So it's really kind of hard to judge, Vincent, everything about this. I mean, I think positives are positives. I think it looks nice. I mean, but to judging what the titers mean, and then as you're saying, to understand what it means in terms of protection, tricky. And then you also can't use NEUT as the only standard for a protective antibody for lots of reasons. First of all, neutralization in vitro is not neutralization in vivo. Certainly neutralization of a reporter virus is not neutralization of the real McCoy. But then on top of that, there are all sorts of antibodies like for flu that we know about now that, you could class them as neutralizing in vitro depending on how clever your assays are. But they were certainly missed in the past, completely missed by including me and other people, that in vivo function quite well because the blocking infection, the only way to do that is not simply blocking attachment. There's other ways to block infection, and antibodies can work even by not blocking infection, depending on what the virus is and what the circumstances are, by innate immune cells that can use the antibodies to collect the virus, phagocytosis, and destroy it, or innate immune cells that use the antibody to bind the infected cell surface and kill the cell. So, again, knowing what's protective, even when it's just quote unquote, just antibodies, is not trivial in vivo, even when you have a really good animal model. So, but this is the first step. A really important question here, and what's present in this paper and particularly the next paper is there are people who are clearly infected, people who are really sick, who don't make antibodies. And what? As a viral immunologist, I'm like, no, that would never happen. But that is clearly happening. And then, I'm sure they make some antibody because it's always a matter of sensitivity. Unless they're B cell deficient, they're going to make some antibody. They have to. But, at what level? And then why is there such a, how could you be so sick with such a huge virus load and not have a better antibody response? I think that's really interesting.
All right, we'll get to that. But what you just said about neutralization. So what is a lot of companies are making neutralizing monoclonal antibodies for treating people, right?
Right.
So
yes.
What you're saying is saying that's not the only thing you should be treating them with. Is that right?
Oh, no, I'm not saying that. I'm just saying there are other antibodies that could work. Right. And you're probably, gosh, well, okay. So let's get real. The whole use of stem antibodies as passive immunity for flu are exactly those kinds of antibodies. They do not work very well in a classical neutralization test, but in vivo in animals, they work quite well. Then in humans, I'm not sure what's been published yet, but what I've heard through the grapevine is they're not working, the stem antibodies. But there are certainly going to be antibodies like that for this virus, and maybe even more of them, because for attachment protein for a virus, this is gargantuan, and there are just acres of epitopes on this on the surface of this protein that you just figure, man, what are antibodies going to do when they bind there, right? How functional could they be? They're so far away from the receptor binding domain. And there's going to be a lot of interesting biology to figure out about what these antibodies do. And, and it's a virus. It's just like daring you, it's just like, oh, I don't care. Here, just flaunting all these epitopes, and not quite so simply, but because there are a lot of sugars around, right? And the oligosaccharides can really mask the epitopes, but the density is nowhere near what it is for HIV. And even with HIV, with GP160, there are still vulnerable, exposed polypeptide sequences that antibodies can see. So this guy's got a lot of sugars, but there's still acres and acres of epitopes available.
What I'm getting at is that these companies are using in vitro neutralization assays in cell culture to identify these antibodies, and then they bring them through animals, and then they go into people. And what you're saying is there might be others that could be therapeutically useful, not based on a neutralization assay, right?
Oh, absolutely. But I wouldn't start with those. What they're doing is the exact rational way to do it. And with flu, what we know, and I think for other viruses as well, is that the more potent an antibody is at neutralizing in vitro, the more likely it is to protect an animal in vivo. It's certainly not worse. Let's put it that way. There's not an inverse correlation. So if you're going to start, that's exactly where you're going to start. So I think that's completely rational. And I would as well I would pick antibodies targeting the receptor binding domain for sure.
Yeah, it's a great first step, but it doesn't mean that they are identifying every antibody that could be helpful.
I guess what I found interesting in this paper, and it's simply that these batches of serum, they had this donor IgG, and then they had separate 50 samples from HIV patients from 2008 to 2011, and none of these reacted with the spike. And to me, it means this virus has not been around before, which we know. And I just think
we know that
this is an interesting way.
Yeah, well, that's why it was created in the lab in the first place.
That's right. That's right.
Oh.
That's right.
A joke, people.
Yeah, right.
Yeah,
that's a joke. It was not created in a lab.
All right. Yeah. So let's move to the third paper then. This is an in-press Nature, which with an impressive cast of authors from Rockefeller mainly and Caltech. And the first author is David Robiani, and the last author is Michel Nussenzweig. And we have here people like Charlie Rice and Theodora Hatzianu, Pamela Bjorkman, Paul Benash. Those are the names I recognize.
Yeah.
Quite interesting.
Hall of Fame.
Hall of Fame.
Yeah, totally Hall of Fame. And I think that they should also get a big pat on the back for this paper, because in this paper, they say that they got all of their participants between April 1st and May 8th. And there is a stunning amount of work in this study.
Oh, you got 50
that
people.
Apparently was done in a month.
You got 50 people.
Yeah. And well said, Brianna. And what I love about it as well is it tells you, so this is good for PIs because there's even though they had all the time to do the work, they apparently didn't have any time to write a discussion. So, because, the writing is really limiting, right? So I think because I know people in my lab wonder what I do all day. So this is just to, hey, it's not so easy to write a discussion.
So let me summarize this. It had 149 convalescent individuals, 7% hospitalized, 78% had IgG to the receptor binding domain, 70% to the spike by ELISA. Okay. So as John would say, the others must have had something, not zero, but they're saying zero. And then they did neutralization assays using VSV spike pseudotype.
They also did a real virus in some cases.
And they say
I'm pretty
neutralization.
Sure. I think that's right.
Neutralization is generally low. 33% undetectable neutralization antibodies, 79% below 1,000, and only 2 above 5,000. So this is what you were talking about before, John. They're infected. They have to be making antibodies, right?
Yeah, I don't know what I know. And I saw the titers and I'm like, they're really low. And so I emailed one of a really good person in my lab who works on flu neutralizing antibodies. I said, Eva, what is the ratio of ELISA titers to Newt titers in our lab with flu? Because I'm really surprised that, so the way you express an ELISA titer can be IC50. So the amount it takes to get half maximal binding for monoclonal, then that also tells you the avidity of the antibody. And for Newt titers, it's the amount of antibody it takes to neutralize half of the virus. And that depends on occupancy. But, the occupancy, what's the rule of thumb, I think, is 80% occupancy is what it takes to neutralize a virus. Dennis Burton has this amazing paper where they show across all the viruses, it's a similar fraction. Mm-hmm. So you'd think the law of mass action would be pretty consistent between an ELISA and a Newt. And I don't think it is actually, even, with flu. And I think that's really interesting about why you have this huge difference in titers. And, I was also, then I went back in literature. We don't work on humans. And I was just interested in, is this particularly a crappy response compared to another primary infection or a primary vaccination in terms of an ELISA titer? Because it seemed really low to me. But looking through literature, it looks like that's not unusually low, where things are, 1 to 1,000, 1 to a few thousand, 1 in 500. So it actually looks like for a lot of the people, they make a pretty average antibody response. And then I think what's really interesting, as you were getting at, Vincent, is the non-detectables, because that's the word they use, which is good. Not that they didn't make antibody, they didn't make detectable responses. And, a guy, Mike Minna, have you had him on TWiV from Harvard? He's great young scientist. Yeah, and he had this amazingly cool paper.
Yes, we did. Yes, the immune memory erasure by measles, right?
Yeah.
Of course.
And then you see this and you think, man, is this virus doing something to B cells? Right? And maybe only in a fraction of the patients. Because these are people who are, well infected, and I was surprised. I am surprised / shocked. And there are other ELISA papers where they have a lot of patients, and 10 to 20% have a non-detectable ELISA titer. And, that alarm bells are going off in my head that this is weird and probably interesting.
So in the first paper, there were Tcell responses in all of the patients. And in this paper, there's some patients who do not make detectable antibody. In your mind, is there a discrepancy between those two observations?
I think it's thresholds. Okay. Possibly, Rich. I think possibly, but I bet it's thresholds. And I'm surprised that. Have you guys seen a single paper where people did Western blots?
No.
Yeah, why is that?
Takes
longer. Isn't that the standard? It wasn't. Remember when HIV got going and you didn't have anything, and for many years that was the standard, right? Was your titer in a Western blot?
Mm-hmm.
So that the sensitivity of Western blots can be really, really good. So I'm surprised that nobody has done that, and then. Also, to the extent that you were making antibody, if you do a Western blot, maybe the problem is no one can get infected cell lysates, right? Because of the BSL-3 nature.
Yeah, that makes sense.
Right? Even though once you pull them out of the, for a Western blot, you could just make a huge batch of infected cells, do the lysis, just boil them in sample buffer, and then you have a supply for a lot of labs. But it would be nice if someone made that commercially available, right? So that everyone could do Western blots, because not only do you have then just one protein that you managed to express, you've got every abundant viral protein in an infected cell. So you've got another probably 10 or more targets that you could detect antibodies to. And then you may also find, I'm sure you'll find some cool things about certain patients responding to certain proteins and not. So, again, early days here, but, if there's no one out there doing Western blots, that's certainly something that could be done.
All right. So what you're saying is that maybe the assays are not informing us properly.
Well, it means something that the distribution means something interesting. It's just what you guys were saying with the infectious, with the RNA persistence, which I'd like to talk about at some point. And I think you pointed out that, who was it? Diane Griffith, pointed out that a lot of what's going on here, it's just because everyone's focusing on this one virus and no one ever looked with other viruses. So, I'd like to, you really, to understand the meaning of the findings with the COVID-2, you have to put it in perspective of the initial findings with other viruses where you had a similar amount of reagents. To see how it really compares, right? And things that look strange here, a lot of that could be, we're just looking at this with such, 4,000 papers a week. I mean, when have people ever looked as intensely at any one thing? Even P53 never got this kind of focus from the world.
Even SV40, T antigen.
Even T, which I even worked on. Yes, exactly. Right, Vincent. Right. Yeah, not even that got this kind of focus. So things that look weird here are probably not necessarily weird.
That's a good point. I appreciate that. Yeah.
Yeah.
What I think is interesting. Let me ask you this, John. So these people that have undetectable newts, do you think they would be reinfected? And if so, would they get serious or not serious disease? It's a tough question, I know.
No, that's a good question. I don't know. So the obvious answer is I don't know, but let me guess. I would guess that they're going to be immune. Show me until shown otherwise. I'm going to assume that the immunity to this is like the other coronaviruses.
Yeah.
Right. That you could be reinfected within a year, on average, something like that. And then the thing we don't know here is that. So here's a question. Are there cases of adults who are infected with the circulating coronaviruses who were not infected by kids? And in that circumstance, do they have a similar, do what is it? What's the percent of severe disease? Is that similar in an adult who had never seen the other coronavirus?
I think that's
right.
So that's good, but
so that there may be patients out there who are seronegative to the other coronaviruses who have seroconverted, and you wouldn't know that before they started running these surveys, like Daniel has been talking about, Dan Griffin, where they run these testing surveys. But they probably now have, with the surveys being in routine use, they probably can identify people, if they exist, who have not been exposed to the seasonal coronaviruses. And then that would be an amazing study to see what their experience is when they do get infected. And probably that won't happen before we have experience with this one. But the $64,000 question here, of course, is when you are reinfected, because that's almost certainly going to happen, is it going to be a milder disease?
Yeah, sure.
Right.
Sure.
We're all hoping it will be, but we don't know that. And if it isn't, oh man.
Yeah, then we're in trouble.
We're in big, big trouble. And at that point, if we don't have antivirals that are effective and routinely available, we're in major, major difficulty.
Do you think that we can learn anything from their data that says that even though there are patients who don't have good neutralizing antibodies, B cells that could make those antibody specificities can still be detected, and so potentially those B cells could be boosted upon reinfection?
Yeah, Brianne, that's brilliant, because that's the point of this paper. Exactly right. And that is, it's a great paper. It is a great paper, because that, I don't know where people have really made that point before like this. And so that's a fantastic point. And their point is that this could be maybe the first example where vaccination is better than infection for providing immunity, right? Because that's what their point is that, listen, these people they're not making a great antibody response, but the B cells are there. And if we design our immunogen properly, then we can do better than natural infection. And, that I think would be the point of the the respiratory syncytial virus vaccine, that Barney Graham's lab has done so much work on. And the point there being that natural infection is not great and we can do better with vaccination. I don't think they know that yet. And these would be first in class, I think, where vaccination is so much superior to infection, and it's entirely possible.
So it would.
What a fascinating concept.
Would be in line with your idea earlier that maybe this virus is messing with B cells, and so a vaccine wouldn't do that. And so that's why these B cells are rare, the ones that Brianne mentioned that produce potent neutralizing antibodies.
The other striking feature in this paper is they get the same antibody sequences out of different people.
Mm-hmm. Yeah, that was really impressive.
And they cite other papers where that happens, but that's pretty unusual.
So what does that mean, John?
What that means is that there's probably not a lot of ways that antibodies could bind the virus, and so the antibodies have to converge on a common strategy.
But you said it was a playing field, these spike. It was huge.
I know, yeah. So everything I'm saying, I'm going to contradict myself because,
Could it also mean that sort of, there's a selective advantage to having these responses, or there's something that's about this virus that's driving that particular mutation?
I don't know what that would be.
I didn't say I knew what it was.
Yeah, I mean, how they're B cells. They just are responding. They're competing with each other, and I think that's an amazing finding.
So I think let me explain this to our listeners, because we didn't really. Yeah. So they took B cells from some of these patients and cloned out a ton of heavy chain IgG heavy and light chains and made antibodies, right?
And this was in both the people who had a lot of antibodies and the people who had lower amounts of antibodies. Right.
But did they do it from anyone with undetectable? I don't remember. They isolated it from 2 top newts and 4 high to intermediate. Okay, so no undetectable patients, right? That would have been interesting, right, to see if they got. Anyway, they find some highly potent neutralizing antibodies in these rare B cells, right? Mm-hmm. And I would have been interested to see if they could get it from someone where they had undetectable neutralizing antibodies.
Yeah, and these are, this is a primary infection, but the affinities that they have of these monoclonals are really, really high, and which is really kind of interesting, actually. They're as good as you get almost with monoclonal antibodies. So, and it didn't take a lot of, okay, so just to go a couple points, Vincent, because the listeners may not understand this. So, the B cell repertoire is not fixed. And there are all sorts of different, there's alleles so the B cells the antibodies are created by mix and matching of domains. It's like the T cell receptors. They're quite remarkable. So no two people, even if they're identical twins, have the same antibody repertoire. The antibodies in the naive repertoire, the so-called germline, they're made by mix and matching bits. And even if you mix and match the same bits, they're not necessarily identical, because when they're mixed and matched, there are amino acids that are filled in, and there's an enzyme that fills in as well. Blanking out on the name of this enzyme. So the antibody repertoires are, yeah, exactly. Thanks, Brianne. So the antibodies they're different between every individual. And then once the B cells get activated, they're more different because they get somatic hypermutation, where the B cells deliberately mutate the parts of the immunoglobulin that interact with the antigen, and then selection goes on for antibodies that bind better. And what they found then is that from a bunch of different, like 5 different individuals, they found basically the same antibody sequence, which means that they're binding the same epitope on the spike protein. But it's also quite remarkable because just from chance, you wouldn't expect that. And so they're calling this convergent evolution, where, you have the same kind of goal in mind. And so, a penguin has fins like a fish does, right? But from a whole different way of going about it, the convergent evolution, because they both have the similar problem of propelling themselves through the water. And so in this case, you have antibodies that need to bind this thing. And instead of coming up with their own solution, which is typical, very typical, you have a bunch of antibodies that have the identical variable region, which is, if not entirely unprecedented, highly unexpected and really cool, and bears deep thought about what that means. And you guys have covered some of the basis there. And yeah, I mean, it's the exact opposite of what I was saying, of course, but I don't know. Brianne, so, and so what everyone always falls to is the virus wants to do this, and it's tricking the immune system by this and forcing the immune system into the immunodominant. And I really doubt that, but whatever, whatever the idea. Yeah. Well, whatever. I don't care what the hypothesis is, but it should be something you could test and just do experiments, right? Mm-hmm. Because that's all you need is an excuse for doing experiments. And then the truth often falls out of just, it's completely the opposite of what you thought, but it just took a creative experiment to open the window to make that discovery. So it doesn't matter what the hypotheses are, listeners, just do experiments that are reasonable. Okay. And then you might find something. And, all of us know this, when you disprove your hypothesis and find something else, it's always better anyway.
Yeah. Being wrong is just as important as being right. Actually.
It's way better, Rich, being wrong, actually. Right. Because then you've actually learned something new. Right.
Right.
Yeah.
Yeah, I would point listeners to figure 3D, because every time I look at that data, I just sort of shake my head and am shocked by how similar these antibody sequences are.
That's cool. This is a great thing.
Let me go back to this low, undetectable, low NEUT. So we explored this idea that the virus's infection is messing with this, and the consequence is reinfection, but maybe you get milder disease like the common colds, in which case, okay, so why is it milder? Could T cells be contributing to making it milder? CD8s, could they limit reinfection?
We don't know.
It could be though, right?
Could be. And there's no human respiratory virus where we really know the importance of the CD8 T cell response. There's good evidence, reasonable evidence in flu. Like there's this really nice paper from 2013 that the SETE paper cites that there's a really nice correlation with how well people did with the pandemic flu in 2009 based on their CD T cell response. But the evidence is really thin. So we don't, and I'm not saying they're not. I'm just saying we don't really know. And so, Vincent, absolutely, they could be really important. And the beauty of the vaccines that are nucleic acid based is they were there, as Rich, as you've been saying, it is true. If you have a pure protein vaccine, particularly with a naive individual, you will not get a detectable T cell response from everything we know. Okay, so if you take an inactivated virus or a protein, and a lot of the how many vaccines are there, guys, that are being in trials? 30
There's
something, right?
There's 8 in trials, but there's another
30 on the and so there are a bunch of protein-based vaccines, which is the old way, standard way of doing it. These are not going to give you a CD8 response, and they, in theory, then are not going to provide the kind of broad protection. And for all we know, Vincent, it's the CD8s that give you the mild infection, and maybe it's the resident memory cells that make it more mild. And their resident memory cells, oh man, measuring that in a human being, people are not really keen to give up, bronchoscopy specimens. No. Yeah. So this stuff is really, really hard to figure out in people.
Yeah, I would love to at some point see what kind of data Moderna has on T cell responses in addition to their neutralizing antibody responses.
You have to assume they're doing those experiments.
I assume they are. I just haven't seen any data, and I like data.
I don't know, Rich. I mean, the VRC is, in as much as the VRC is involved, yeah, because these guys are the best in the world at those kinds of assays. I mean, they do them, they could do 100 colors at once, right? It's quite incredible. So I think so. Yeah, you think they're measuring CD8.
So is it fair to say that doing the T cell assays is is more difficult than looking for antibody?
Oh yeah,
right.
Yeah, yes. A high school class could look for antibody by ELISA. It takes a big lab. Well, nice to have a flow cytometer and expertise, and the T cell element of the antiviral response has always been really daunting for characterizing immunity to viruses. And the viral immunology field, it's not a big field because you have to have this combined expertise in immunology and virology, and to put that in one person is not easy, and then often it's a collaborative effort where you have the virologists working together with the immunologists. And one of the more positive things that have happened in science in the last half generation is there is a lot more collaboration now and labs working together, and it's reflected in these multi-authorships on papers where it really does take a village to understand what's going on. And now that we know how we're getting a glimpse at how complicated the immune system is, nobody could be an expert in all these aspects. And if you want a comprehensive examination of what's going on with an antiviral response, you have to recruit all sorts of expertise. And, I don't know, there hasn't been any NK papers yet either, right? So
no, I don't think so.
Yeah. I mean, that's, as I was saying last time, man, that's Byzantine. So, and important. And so it's, silver lining, man, we're learning a lot. We're going to be learning a lot of viral immunology thanks to COVID-2, just as we have because of HIV, which really accelerated progress in immunology in the late 80s and 90s. I mean, most of the resources for our viral immunology went to HIV. It recruited a lot of good people, and they made all sorts of advances. And things like this give a real shot to a field, and the field makes a major advance. And it's just great because, thanks to HIV, we were certainly much better prepared, at least scientifically, for this virus, if not at the level of boots on the ground where we were not prepared at all. But scientifically, man, we're ready for this, right?
There's been a lot of flu immunology too, right?
Yeah. There have been. And, certainly the basic work, most of the progress in the basic understanding of the immune response has come from the animal models. And flu for the CD8 T cells has been the major model, but the LCMV people get a lot of credit. And, it's cool because, like the checkpoint inhibitors, a lot of that work for cancer, a lot of that work was done by viral immunologists, Rafi Ahmed and others. And, it's the immune system. And there are unique aspects of viral immunity and tumor immunity, but there's a much bigger aspect of shared mechanisms. And so each field teaches each other field. And for young scientists out there, you should be reading broadly. That's the message here, is that the broader you can be, the more likely you are to think of good experiments and make discoveries. So breadth is good, and talking to other people and collaborating is really good.
You asked earlier who would want to be an immunologist, and I think that all of those things are reasons why someone would want to be an immunologist, because it's just so cool.
Yeah, it's amazing.
I was kind of joking.
I mean, Vincent was joking, but just the creation of the T cell receptor and the immunoglobulin, it's amazing.
Yeah. Well, I think in a similar way, virology and virologists, viruses are cool too, but I just don't think we say that it's complicated as much as immunologists do. That's all.
Well, once you get into the cell biology, it's every bit. Of course, the beautiful part of virus is how relatively simple they are, right? So no question. And,
compared to cells.
So finding viral peptides, it's shooting whales in a barrel. Finding tumor peptides, it's really, really hard. So, all sorts of things viruses make easier. But at the same time, the virus is the cell. It's just a different program. And if you want to understand how a virus works, you then have to understand all of cell biology. And good luck with that, Vincent, right? I mean, that's as complicated as it gets.
Let me ask you a experiment question. Is this possible? Could you take, say, a COVID-19 patient and get T cells out of them and see if those would lyse virus-infected cells in culture? Is that a doable experiment?
Yeah, they will. Yeah, you could do that. The trick is BSL-3.
Yeah, of course. But someday this will.
And so that's the problem. Yeah, I mean, but there are other, you could look at whether they're expressing the lytic protein. So there are markers on the T cells that would tell you very accurately predict how cytolytic they are.
I see. Okay.
Yeah, so you could do that without actually having to do a killing assay. Mm-hmm. But you wouldn't even have to do that. So sorry, Vincent. You could test their lytic activity on peptide sensitized targets once you have that, or target cells expressing the protein from other vectors, right? So, that's how Jack Bennick and I got started. In the early 80s, we had first collaboration with a really good vector guy was Bernie Moss, who in the end, Jeff Smith was a postdoc at that time. With Bernie, Jeff put in all 10 of the defined flu gene products, and we were the first ones then, just using that relatively simple system, to map the immune response at the level of individual proteins with these, clone proteins. So, Vincent, that could be done immediately. And but there's really, if you can measure a T cell activity by cytokines, I'm sure there's an example somewhere, but there's usually a pretty constant fraction of those cells that are lytic, which is most of them.
Yeah.
So, yeah.
I mean, it seems to me that just based on our little discussion here, there's a ton to do. And I wonder with SARS-CoV-2 and coronaviruses in general, and, they haven't got a lot of attention in the scheme of the world, right? As far as infectious disease go. And I wonder if this, when this pandemic comes to an end, we have vaccines that are working, if people will continue to work on these questions, because they seem to me fundamental immunology questions, or whether they'll just forget it and move on to others. Like all these new groups, Nussenzweig didn't work on coronaviruses before. Will he continue? And all the people that are...
Well, but he's been working on viral antibodies for quite a time and made, arguably among the best contributions to the HIV antibodies. So this is right in Michelle's wheelhouse. And for the kind of work that Michelle likes to do, or I like to do, the virus is immaterial. You want to make basic discoveries about how the immune system works. And here, the basic discovery is that, we get this convergence and, everyone makes decent antibodies. So you could make decent antibodies. So I think, if you're a basic scientist, that's really more of what your goal is, rather than coming up with the vaccine or the treatment. You want to understand things, and then other people will apply those things. And you know how it works. I mean, once the polio vaccine was there, this huge field of virology and, vibrant, it kind of got, clipped, right?
For sure.
Extremely.
Sure.
And that's a shame, because polio virus, it's a great experimental system, and every virus will teach you lessons about cell biology, about immunology that you would really like to know. But just speaking of which, so we don't know that we don't know what, I can't find a single paper about what coronavirus infection does to levels of class 1 on the cell surface. So for all we know, CoV-2 has proteins, some of these non-structural proteins, for example, that are interfering with antigen presentation, right? We don't know that. So we're working on that now. Okay. And, it's a cool thing to do. And we'll continue that even if there's a vaccine tomorrow, because I agree with you. It's an interesting question. And, we've basically studied drips in 2 systems, and I'd love to expand it to other systems to see if there are exceptions. What are the exceptions? And so this will definitely, some of the research will absolutely continue onto coronavirus. But of course, it won't be at this level of intense interest that we have now. And but yeah, you're absolutely right.
I also think that if it remains a BSL-3 virus, that will have a chilling effect. But if it's reclassified at some point, which it could be, that would really help, right?
Yeah, the whole philosophy of BSL-3, I mean, you could get it on the Metro to work, right? Which is the BSL-0, but then in the lab, it's a BSL-3. And I don't really completely understand that. I mean, have you ever known anybody to infect themselves with a BSL-2 virus in your own personal experience, guys, as virologists? I mean,
yes.
Okay.
With some questionable lab practices, yes.
Okay,
Right.
But this could certainly be a BSL-2 plus, right?
Yes.
And then it would facilitate research. And it's just like everything else. We could have complete isolation and destroy the economy. We could make this virus so hard to work on that no one could work on the virus. And what you're always looking for is the happy medium, the compromise where it's safe. It's not perfectly safe, but it's safe enough. And again, this is not Ebola where you're going to introduce a virus that isn't already present in a large number of people in the population. This is a virus that's circulating. So why don't we have BSL-3 on the subway then? I mean, that would be ridiculous, obviously. But I think to have it in the lab, to me, this is, I'm not sure this is really necessary. And, like a bird flu, I mean, we've tried to do some experiments with some proteins from avian flu. And I've always found it ironic that the virus is walking in front of me with the geese and in front of our building or the ducks, and yet we can't work on the virus in the laboratory. So if I make a misstep and I step in the goose in the goose poop, there I've got H5N N2, but in the lab I can't work with it. So it's just like, really, I understand that we have to be safe, but you're also by slowing down progress, you're also costing lives on the outside. And I think the balance for this virus is I don't really understand why it has to be a BSL-3 virus.
Well, West Nile is a BSL-3, and it's a mosquito-transmitted virus.
Yeah.
Why is it a BSL-3, right?
I don't understand. There's a lot of CYA in all of this, right?
Well, it's a historical reason because years in the 50s, I think people were injecting the virus into tumors to try and get rid of the tumors. And in a few people, it went into their brain and caused severe neurological problems. But, you have to inject it into the person to do that. So that's why it was classified BSL-3. Yeah. I mean, but John, this whole discussion it's also, this moratorium that we had a few years ago on gain of function. I mean, this is all limiting research, right?
Well, I would like those people to say, I'm sorry to the world, because you can look at Ralph Baric, when the ban was happening, they let people appeal and make a case. And Ralph Baric had this wonderful thing. It's online. It's a document that you can see. It's public information, where he said that basically, well, you could ban this research, but in nature, the coronaviruses aren't going to listen to this. That's right. And that's exactly what happened. And so I would like these guys to come forward with a mea culpa and said, listen, we were wrong. It's much more dangerous to have ignorance than to prevent, than the slight risk there would be of creating something.
They will never do that. They will never do that.
Well, I'm not sure. I mean, I think they should be given the opportunity, and I would love to hear that, because these are intelligent, thoughtful people who I think had no agenda. They were just concerned, and I think their concerns were wrong. And it was striking to me that none of the major players, they were actually people who were working on the viruses that they were trying to regulate the research for. Right. And so that's a problem, because if we don't trust the experts, who do we trust?
You're absolutely right. The problem is, Richard E. Bright, you know that name?
No.
He's one of the group with Mark Lipsitch and some other people who are vehemently opposed to gain of function, and they're responsible for the moratorium. E. Bright, he's a professor at Rutgers. He's saying this thing leaked out of a lab because they shouldn't be working with dangerous viruses. I mean, this is the kind of crap, and
it's just. So, I mean, that, first of all, there's no evidence supporting that. And second, even if that was true, it would still not be a useful policy. To prevent diseases, you have to understand them and to work on them rapidly and make progress. The more understanding you have as a baseline, the more rapid the progress is going to be. And so you just, you have to work with viruses, under safe conditions, but where you're actually working with them. And for viruses, the only thing that they're selected for is transmission. And so then you have to work on transmission at some level to understand the biology of the virus. And to prevent that research, you're just hampering humanity's ability to deal with a pandemic. And I'm not going to say that, we would have been able to deal with this had Ralph Barrick been able to do with experiments, but the principle is still there. It is certainly possible we would be better equipped to deal with the pandemic if the research had been allowed to go on.
I fully agree. And you're preaching to us.
I know I'm preaching to the choir, but maybe there are people out there who, sure, particularly people who would go into public policy who need to hear this argument.
Mm-hmm. All right.
Let me just get back to one thing before I'm off, Vincent. So this persistence of the RNA by PCR without the virus, that I think is really, really interesting. You guys have talked about this, but everything I know about RNA says it's incredibly labile. So I do not understand how you can have persistent RNA without constant production of RNA.
Yeah, well,
so I think that RNA there have got to be cells that are producing the RNA. And okay, let me just say something completely crazy, right? That, the most logical possibility is you have cells that are infected and they just aren't producing infectious virus that you can detect. Okay. But the second possibility would be that there's some sort of CRISPR-like system going on where some host cell has captured the nucleic acid information, and then for whatever reason, it is producing the RNA to keep the T cells active, something else. But you cannot be detecting RNA without some active production of RNA. And so I think in those patients, it's really important to know what the source of that RNA is.
Well, it's not just this virus, it's many other infections that
exactly, whatever. And I think I mentioned this last time, there was this paper from a Brazilian group where they found evidence that flu was replicating in the lymph nodes of kids that had recovered from flu months before. And no one ever knew that flu could make this persistent infection if that was what was going on. And so that, even though you think a virus isn't persistent, we don't really know that. And for this virus, we don't know that. And it looks like in a lot of patients, the virus is persistent, right? People are sick now for, as Daniel was saying, for 8 weeks, right? Turning PCR doesn't go negative. So the virus really could be replicating somewhere. I think like one of the really interesting features of this virus is that, all of these non-lung symptoms that certainly imply that there's infections that are going elsewhere, which implies that there's a viremia that carries the virus there. And, the way that the effective vaccines work is viruses that have a viremic phase. Mm-hmm. And wouldn't it be cool if the coronavirus vaccines for this virus work? They don't do so great about the lung infections, the upper respiratory part, but what they're really good at, the vaccines, is providing what results from the viremia, which maybe in the end is an important part of the disease process. So maybe with vaccination, we still have even some lung infection, but we don't have what appear to be the difficulties with the circulatory system and kidney failure and digestive and all that the vaccine is really good for. So it's just so much to learn with this virus.
We talked about that last time, this idea that, viremic viruses like measles, mumps, rubella, they give you really durable immunity. Yes. But the wimpy respiratory viruses that don't have viremia do not. Maybe the key is to make a vaccine that has a little viremia, right? That's what OPV does. That's maybe does V.
Oh, you mean for a live vaccine?
Yeah, for an attenuated vaccine, maybe.
Yeah.
I don't know.
Yeah, I think that some of the questions that have been raised about coronavirus immunology having to do with duration of immune responses and duration of protection or cross-reactivity will give immunologists things to study for many years to come.
Oh, absolutely.
The question is, why weren't they studying it before?
Well, it's always funding, Vincent.
I know, of course.
I have a whole PhD dissertation.
Yeah, I know.
If NIH builds it, they will come.
I agree.
It's lab of dreams.
SARS-1 was very interesting, and it should have been worked on. And, a few people did a little bit of immunology, but not much. And, yeah, it's all funding. I agree. But, boy, we could have been way ahead of the game if we had been working.
We could have been. But, again, the BSL-3 is a real turnoff.
Yeah,
my guess is that the research on this is not going to die off real quick. I would hope that after 3 spillovers of coronaviruses into humans over a period of less than 20 years, that people would get the message, in particular,
Rich,
given this one.
Rich, you are absolutely right. And, if we get a vaccine for this one, there'll be another one.
Yeah.
You're absolutely right. And the coronavirus is on the top of the list.
SARS.
Zoonotic viruses that can jump.
SARS-1, the problem with that is not only is it BSL-3, but it's a select agent. You have to have FBI clearance to work on it.
It's insane.
Crazy.
Yeah. I mean, the idea that you can keep track of every microliter, this is only the idea of a crazy person. Really. I mean, anybody who's worked in a lab knows you can't do that anyway. But if someone was intent on stealing the virus, they would take a microliter out and put a microliter back in. I mean, come on. Mm-hmm. It's impossible. I mean, the whole idea of the select agent, the level of scrutiny that they give, if it's to make you afraid, okay, I guess that's a psychological effect, but the practical security that's afforded is it's a fantasy. It's a fantasy. And the cost, again, okay, so we have a policy. It could do this, but what are the costs of this policy? And that's what's so hard to factor in, right? What are the costs of ignorance? They're incalculable.
Yeah,
right.
Totally.
They're calculable. And let's say that, okay, in this case, right. So the immunology, we weren't going to have a vaccine because we didn't know what it was, but we certainly could have had drugs. And there were people out there like Ralph and Matt Freeman who were begging for money to do drug screens for coronaviruses. And you guys have had, you've had shows on this, great shows about the targets. There are a gazillion targets in this virus for drugs, right? And yeah, a drug is not as good as a vaccine. No, it's not as good, but it is better than not having a drug. And it would have saved a lot of lives already. So, man, that is a huge cost of the government just not paying attention, and not just our government, every government in the world. And we're just, again, I'll end where I started. We have to get smarter as a species. We really do. We have to think harder about what the real threats are to society and how we should spend our resources accordingly. Should we spend them on a wall, or should we spend them on developing drugs for viruses and pathogens that could kill us? Really,
seems like
right? $15 billion for something that won't do nothing.
Like an easy
answer
to that one.
Yeah, it's an easy answer, but people vote. Young people vote.
All right, that's a good way to end this. Let's wrap it up. This is TWiV 620, microbe.tv/TWiV. Questions and comments, TWiV@microbe.tv. Consider supporting us in our work, microbe.tv/contribute. Our guest today from NIH, Jon Yewdell. Thanks for coming back.
A pleasure. And let me just mention that all of the opinions I've expressed, which are many, are my own and do not reflect any of the opinions of my employer.
Of course, it's all our opinions, and we had a good discussion today. It was fun.
Excellent. Yeah. Super fun.
Yeah,
Rich Condit, Emeritus Professor, University of Florida, Gainesville, currently in Austin, Texas. Thank you, Rich.
Sure enough, always a good time. And once again, to me, it's a privilege to be a part of these discussions and get to ask the questions that I have on my mind. And of course, it'll be past this by the time this episode drops, but in about 45 minutes, they're scheduled to launch the SpaceX Dragon with pilots, with astronauts on board. And I'm going to go watch that. I recommend it.
Yeah. Thanks, Rich, for the reminder. I'm going to watch.
Brianne Barker's at Drew University over on Twitter, BioProf Barker. Thank you, Brianne.
Thanks. It was great to be here, and it was really wonderful to have you, Jon. Thank you so much.
Oh, pleasure. Good to meet you.
Good to meet you, too.
Glad you didn't have to leave, Brianne. It was worth it, right?
I am, too. I have enjoyed Jon's papers for a long time, so getting to sort of meet him in this way is really exciting.
I'm Vincent Racaniello. You can find me at virology.blog. Thanks to the American Society for Virology and the American Society for Microbiology for their support of TWiV, and Ronald Jenkins for the music. This episode of TWiV was recorded, edited, and posted by me, Vincent Racaniello. You've been listening to This Week in Virology. Thanks for joining us. We'll be back next week. Another TWiV is viral.